to talk to you about trees, and I have no idea how trees work. That's all the fun, right? If I knew how trees worked, this would not be very, very interesting scholarship I am doing here. So we're going to talk about things we don't know about trees, because that's where it gets really cool. And we might think of trees as something that are pretty well studied, like especially something like redwoods, but there's really so many things we don't know about trees. And as much as I might like to think of trees as a series of tubes and capacitors and resistors, it's nice to put them in the context of the forest. Uh-oh, clicker, click. What am I doing here? Yay, okay, oh, wrong direction, nope. Trees, yes. So it's nice important to remember them within the forest, right? And a lot of my work focuses on trees in the context of their environments. So all of these abiotic factors and everything going on around them, like are they in a hot site? Are they in a dry site? How do they use water? How do they handle things like climate change? And where are they going in this crazy world we have coming and have delivered to the trees here with no fault of their own? So a lot of my research is focusing on the interaction of trees and climate change and feedbacks between trees and climate change. So how does climate change impact trees? And then how are trees in turn impacting climate change? And within this context, we think about all of the different climatic stresses they are facing. So these big, tall trees here are fighting gravity, right? So if we think about the trunk of a tree here, it's really just a series of like little tubes all glued together into one big giant tube. So I like to think of trees as these just giant columns of water, right? Because that's mostly what it is. It's mostly just water in there. There's just a whole lot of water in a big old tube reaching up to the sky. But it is fighting gravity. So this water being there is not just on its own. It has to somehow maintain its connection to itself. So the water molecules have to stay stuck together. Bad things happen, you get air in your system here. And they have to go all the way up over 100 meters, over 110 meters, all the way up to really, like, if we're talking margins, this is like the margins of what is possible for plant life. And as cool as they might be from, oh, wrong direction, from the ground level here, really, they're impressive, they're big, but the business end of the tree is higher up in the canopy. This is where the leaves are. And the leaves are what is doing all of the photosynthesis. But the leaves are also the site of the biggest risk to the tree. So we might think of them as like, you know, they're productive, we need to have them, we cannot be a tree without leaves. But this is also where the climatic stress, things like water stress and heat stress, this is where they might do the most damage to the plant, right? So this is the part that is the most at risk. They're out there on the margins, the little itty bitty, most vulnerable parts of the tree. But they also are what the entire tree is hinging on, right? You can't photosynthesize, you can't grow, you can't do anything without your leaves. But this is where we are at the most risk. And here down in the lower crown, we don't even see leaves in a redwood for the first 50 meters or so. But here down in the lower crown, they might be in really deep shade. But then as we go higher up in the tree, they become increasingly exposed. And so within the crown of the same individual tree, we have completely different climates. So up at the very top of the tree, we might have 100% light availability, all of this wind and just sun exposure. Down at the bottom, we have almost no light. Like they are eating up like 90% of the light by the time it gets to those lower crown leaves. And a very sheltered environment. It might be 20 degrees cooler down there. By the time we get near the top, we start to get at like the real limits of what is possible for plants. So that is why I think trees are so cool, is because this is the limits of what is physically possible for life on Earth, right? We cannot get bigger than this. And when we try and get bigger, the top dies off, right? So we're at these like hard limits in what's going on with the tree. And the leaves here, you can see these leaves in the margins here, they look really different than the redwood leaves we're seeing on the ground, right? So there's a lot of constraints going on, mostly due to water stress. So they can't expand as well at the top of the tree as they can at the bottom, because they're constantly fighting gravity. So the top of the tree is always, always, always under permanent water stress. And that's where it gets really interesting. So we've looked really closely, really closely at these leaves here. And you can see there's this massive gradient in how they change across the crown. So this does not, like if you looked at this, you would not think redwood leaf, right? You've probably seen an awful lot of redwood leaves around here. And this looks really different. And when we look across the whole crown here, we see this incredible diversity in structure and also in function. So if you look at these leaves we have here on the left, there's this big variation in whiteness. These leaves, their whole job is photosynthesis. So these left-hand leaves, we call them peripheral leaves. They're doing all of the photosynthesis of the tree. These other leaves here we have off to the sides, they're only like 5 to 12% of total leaf area, but they are not photosynthetically productive. They're barely even breaking even. In a lot of cases, not breaking even. They're respiring more than they're photosynthesizing. But their entire job on the tree is to absorb water. So they're there, they're leaves, but they're not leaves for photosynthesis. They are leaves that are entirely for water absorption. And even though they're only about 5% of the total leaf area, they do about 30% of the water absorption in a redwood. And it all comes down to these really specialized structures they have. And that's where it starts getting really cool, right? So we've looked pretty closely at the leaves here, but now we have to look really closely and start getting in the cells. So if the leaves are the business end of the tree, the cells are the business end of the leaves. And it's in these tissues here that we start to see really cool variation and differences among species that really impact who are gonna be the winners and losers with climate change, what are the limits to stress tolerance, what's possible for a plant. So in here we see all kinds of crazy things. We've got water tissue, we have airspace, right? To do photosynthesis, you have to get air, you have to get CO2 to your cells in order to absorb it. So the airspace inside a leaf matters a lot. The water storage tissue, which you see these big kind of pink wings off to the side here, that's all really important too. You might be able to store more water in your leaf than you'll use in an entire day in some cases. So looking at these cells here, we have to look even closer. So we've started getting into a lot of anatomical detail and looking at how all of these different structures change with height in the crown, but also change with different environments, and how these relate to differences among species. So over here in panel E, we have the top tissue is water-conducting tissue, and this darker blue tissue is sugar-conducting tissue. So looking at how the ratios of these things change across the tree, right? Is this leaf provisioning a lot of sugar back to the plant, or is it mostly receiving a lot of water from the roots? Like, how is this balance working out here? So looking at all of these things are telling us a lot about how they work. So we measure these things as functional traits. And this lets us get into the physics of the plant here. And we get even closer here. So getting closer and closer and closer, we can see as we start to approach this chloroplast level, all these little green balls in here. So leaves are kind of clear. We don't really think of them as clear. They're like these clear jars full of little green marbles, and that's why they look green to us. The green is in the chloroplast, and the chloroplasts are the business end of the cells here. These are doing the photosynthesis. But in order to photosynthesize, like I said, you have to get the CO2 in there. And so on these tiny levels, it comes down to like biophysics of diffusion moving through here. How close are these individual chloroplasts to the cell wall? How thick is the chloroplast itself? And how far does the air have to diffuse in the liquid phase? So if we're thinking about how air moves through or how CO2 moves through a leaf, it moves 10,000 times faster through airspace than it does through water. As soon as it has to dissolve and move through a liquid cell or move through a chloroplast, it is massively slowed down. And we can use some really cool like physical laws to calculate these things and do all kinds of cool stuff. But to really get at these differences in airspace and see how things are moving, we really need to stop cutting big trees into little pieces, which is a lot of fun and then definitely my jam, and look at them in 3 dimensions. So really look inside the tree while it's still alive and see all the cells and see what's going on. But for that, we definitely need a bigger microscope. So in order to see inside a living thing, we're using CT scans, particle accelerator-based CT scans. And these are really similar to what Allison is showing you here that are produced on campus. But instead of looking at tissues and seeing like, the whole inner ear of a shark, we're getting down to individual cell level here. So this one is at the Swiss Light Source. It's the biggest microscope in the world, which is kind of awesome. And we're doing experiments here to look inside of twigs and leaves as they're being exposed to water. So we're being able to see inside the cells for the first time and see what is actually happening. So there's all kinds of theoretical things and stuff we can calculate, but being able to see what actually goes down has ended up blowing our minds in a lot of ways here. So at this red arrow here, you see that little tiny droplet inside that empty cell here? This is something that is only theoretically possible, right? This is how an air-filled cell from water stress damage refills itself. And being able to see these droplets in here actively forming after we've exposed these to rain really tells us a lot about what this process looks like and how this works, and allows us to take our mathematical models and all our biophysics mumbo-jumbo and relate it to real observable phenomenon going on inside a living intact twig that is not dead. This is still live plant material being scanned live. We're putting it in, a very heavy dose of radiation, but we're only doing it for like 90 seconds, so it stays alive briefly. And we can look at other things like the refilling of cells after exposure to rain, right? So if you are water stressed, you end up with air in your cells as a plant. This is not great because you cannot conduct water past an air-filled cell. It's just like getting an embolism in your vein, right? No bueno. But in plants, after they've been exposed to rain, these cells can refill. And so we're able to look at these things here in the particle accelerator and see them at this submicron resolution and actually watch these processes happen before our eyes. And this has been really, really exciting. We're also doing this work at the Advanced Light Source at Lawrence Berkeley National Laboratory. So this is the world's brightest flashlight, essentially. It is the brightest source of radiation on planet Earth. And they let me push the buttons. It's amazing. So we just found out today that my grad student, Grayson Prater, just got another 24 hours of beam time here on this particle accelerator for spring. So that's really exciting. And when they say 24 hours, they mean like a 24-hour shift where you are just staying up for 24 hours to do all your scans, because this does not shut down for anybody. So you get 24 hours, you are staying up for 24 hours doing your experiment. But it's really cool. And some of the things we're doing here, so this is wood, this is redwood wood, looking inside here. And what we're doing is we're using this like a really fancy densiometer. So foresters have been X-raying wood for like, I don't know, 50, 60 years and being like, this one's denser, this one's less dense. Because what you're seeing in an X-ray is actually the density of electrons. So where it is wider, like brighter in this picture here, is where the electrons are denser. And we can relate this electron density to physical density. And so we can use this to, for the very first time, be like, oh, this cell is what's making this wood denser than this wood. So this is really important for like wood products and wood quality and all kinds of like understanding mechanical stress. But it's also really important because this tells us what is going on with the ratios of these cells and what this might mean for the tree. So all of these ones here that you see that look like little caterpillars, those guys are called rays. And they are in there storing all the starch and all of the sugar. And they're also really heavily involved in carbon storage. And so this work has allowed us to this really interesting observation that it turns out there are two kinds of rays in redwoods. Now bear with me here. We've got this in the purple, we've got what we call short rays, and highlighted in the blue, we've got what we call tall rays, because we're super creative like that over in biology. And what we've found is that these two ray types have completely different densities. In sapwood, which is the living water conducting part of the tree, and in heartwood, which is like the red in the redwood. This is the part that's storing compounds, and it's very involved in like tree longevity and also carbon residency time in the system. So once you lock away carbon in these cells here in the heartwood, that would be the red stuff in the heartwood cells here, this carbon stays in the system for maybe 1,000 years longer than if it is sugar in the sapwood. So it's really interesting stuff for us to be able to pull these things out. These are actually the highest resolution scans of any plant material ever made. Just saying. And so looking at this stuff has really like led us to some kind of cool conceptual things, but also like turned us onto a whole kind of world of possible experiments we could do. And so taking advantage of other kind of modern ways of looking really, really closely at plants, we're using some new tools in fluorescent microscopy. So these are something we've only kind of recently developed of looking at natural plant autofluorescence. So these are this column here under control. That's what colors plants glow under UV light all on their own with no treatment. But we're also using this to use fluorescent tracers and see the pathway of water across the plants. So this is a whole different variety, chalcofluor white, 5,6-carboxyfluorescent. These are different types of chemicals that move through plants in different ways. So one of them moves between the cells, one of them can cross membranes. They do different things, and by using these different properties, we can figure out how does water get from, say, the surface of a twig to the inside? What pathways does it take? What does this look like as it moves through different tissues? And so being able to track this at different stages, right? So maybe I put it in for 20 minutes, I see how far the dye got. I take it out, and I put it in for another 20 minutes, I see how far the dye got. This is letting us build these really interesting biophysical models using both masking and all of these things that are letting us get at these resistors and capacitors and this really fine biophysics of how water is moving through twigs. And this might seem like a bunch of like, wow, okay, that's kind of cool, but we can use this stuff. So now that I know where the different resistors and capacitors are, the things that speed up or slow down water moving into a twig. Now that I know what they are, I can compare these in different species. And I can say, okay, because we have a very thick cortex, we have more hydraulic capacity. Because we have more hydraulic capacity, we will be able to absorb water faster and for a longer period of time. And so I could say that a tree that has a thicker cortex in its little distal most outer twigs, that it would be more capable of taking advantage of a brief rainfall event, and thus probably have more water stress tolerance. So these are really interesting things that we can use to develop like actual real-world hard traits we can go out and measure to compare different trees, figure out who's going to be the winners and losers under climate change, and all kinds of other really kind of fun and wild things we can do with these experiments. And moving on with this fluorescence work, some of my graduate students right now here, this is actually all of this stuff involves my graduate students, but this one in particular is a new method my student Justin has developed to determine the heartwood boundary in redwood and other species. And so he's using fluorescence here, and in this top panel, you can see this yellow and red stuff is heartwood, and the green stuff is sapwood. So by using this natural fluorescence, what he's able to determine is exactly where the boundary is and where it is transitioning. So these little bright green dots in those cells over there, those are chemicals called flavonoids, and those are the precursor chemicals for these red guys here, which are tannins, which is the red part in redwoods. Those are those biocidal compounds. And so by determining this zone, we're going to be able to go in and look at this at different times of year and say, at what time of year do we have flavonoid initiation? How do we see the progress of this heartwood front moving? What do we have differently? So using the actual color spectra of these different things, the reds versus yellows, how does this relate to the actual chemical identity in here? And where are they in the cells? So what is in the water conducting cells versus these rays and all of these like really interesting things? So that's one half of my lab is doing that right now. And we're also using these kind of trait models we develop to look at things like how redwoods absorb water across their entire range. So right now we're collecting redwoods. One of my graduate students, and also we have 62 community college students around California as part of the MESA program, which I'm a MESA alumni at California community colleges as well, and they are collecting on 12 different campuses, redwoods, all over the place, like Pasadena, like we've got them all over the place, San Diego, everywhere, all the way up Mendocino College, like so all over the place, and the students are collecting leaves and they're measuring the waxiness, they're measuring the stomata, and all of these traits that we can use to look at whether or not this leaf will be good at absorbing water. So as you can imagine, a really high wax leaf is not great at absorbing water across its leaf surface. And so what we're doing is we've got these from, I think now we're at like 30 sites we've collected from across the state, and we're looking at the climate of those sites and seeing how the climate impacts these traits that are really critical to whether or not redwoods can absorb water through their leaves. So what we want to know is, as we are losing these leaf wetting events and as fog is disappearing, is the climate pushing redwoods to acclimate in a direction that makes them more able to use fog water, or are we going in the opposite direction? So are the very traits that are making fog disappear pushing redwoods to be less able to utilize it? And so these are some of the kinds of questions we're answering with these trait stuff and with photography at all different levels here. And finally, we're also using this to just find all kinds of strange things we didn't even know. So I don't know what the heck that crystal mumbo jumbo is over there, but it sure is cool, right? And so that's kind of some of the fun of using these emerging technologies, is we just stumble on a lot of really weird stuff that opens up new puzzles and really gets us thinking and is just kind of inspiring us to kind of keep going with this and keep pursuing these things. And now that we have a TEM finally working on campus, we can get into even higher resolution here and start looking. These are the little white rectangles, are individual crystals in the outermost cell layer of the cuticular surface of a redwood leaf, really tiny stuff. But these crystals are awesome because they store a lot of carbon. And when the tree is midsummer, when it can't open its stomata, it can't get that CO2 into those chloroplasts, it can break those back down and re-photosynthesize the CO2 that was used to form them in the first place and keep their apparatus going just a little bit longer. So we're using all of this crazy stuff across all kinds of scales, but still trying to circle back to the tree itself, right? So you can imagine some of these scaling exercises get a little weird, but it's really interesting to be able to look at like these tiny details and then try and bring it back up to what the whole tree is doing. So with that, thank you. And I thank you so much for this award.